First, as a refresher, our biosensor is based on the motion of the molecular pendulum, which is comprised of a DNA linker and bound with an antibody, and this pivots about an electrode surface. When you apply a positive potential, the rigid double-stranded DNA, which is also negatively charged, moves downwards due to electrostatic attraction, and this is against competing hydrodynamic drag. We use chronoamperometry, interrogating the system with a 0 to 500 millivolt step potential, and measure the current response of the system from the attached ferrocene redox reporter. And so this gives us a current response that's equivalent to the probe's time constant. Next, let's look at the experimental probes themselves. Here we have an example of our BNP sensor. So in terms of creating these probes, let's start from the bottom and go to the top. So the bottom where we have our electrodes, so we have fabricated gold 3 electrode arrays, which are either on a silicon or a glass substrate. And from there, we have small apertures of gold where we grow microstructured patterns to increase the surface area of our electrodes. And these have nanostructured features on them. So we use electrochemical deposition techniques for this. As we move forward with the development of our sensor, we are also exploring being able to use just the flat gold surfaces, and we're exploring the sizes that would be necessary for the sensitivity we require. Moving up, we have the probe itself, which is a double-stranded DNA linker. And so these linkers are made from the two single-stranded sequences that we get from IDT. So one of the sequences, one of the strands is a ferrocene conjugated probe that's dial terminated, so it can attach to our gold electrodes. We have two methods of procuring this ferrocene conjugated DNA probe. We can get it commercially from IDT and or biobasic, but we also have in-house methods using click chemistry to conjugate the ferrocene reporters ourselves. Next, we also have our second probe called P2, which is the antibody conjugated probe. This one we do in-house conjugations using an ABCAM conjugation kit. Next, we label the anti-BNP antibody itself. These we get commercially from either ABCAM, GeneTex, and Novus Biologicals. And then finally on top would be the bound protein, the bound BNP that you would see in your solution, and this we get commercially from ABCAM. So to actually create our sensors, we go in the same direction where we first grow our enemies on our electrodes, then we take our conjugated P1 and P2 probes and hybridize them for about 15 minutes, and then we deposit those probes onto our electrode surface, generally incubating overnight. After that, we wash the unbound probes off the surface, and then the system is ready to go for use. So in general, we use gold electrode sensors that are microfabricated on a silicon substrate. So these electrodes have 20 working electrodes. So this is for multiplexing or biological replicates, or for multiple technical replicates. And they also have gold reference and counter electrodes as well as those kind of two gold strips you can see there. So these sensors, when we use them, they're clamped to break out all the different working electrodes for our on-bench setups, as you can see in this bottom clamp here. And in general, for our on-bench setup, actually, we opt to use external counter and reference electrodes using standard materials. So here you can see I'm using a platinum counter and the silver chloride reference electrode, and then we just probe with a wire to all the different pins for working electrodes. And here is an example of when we're actually depositing probes onto our sensors. So we take our solutions and we put them onto the working area of our electrode. And this is put in a petri dish with buffer in a smaller petri dish, just so that the solutions don't evaporate as they incubate, let's say overnight, if we're depositing probes. We are also developing a portable solution for our system for our animal studies that involve continuous monitoring, and for our future commercial applications. So for this, we designed smaller chips. So here you can see there's a chip that's on a glass substrate, again with gold working counter and reference electrodes, and with a clamp for the interconnects. Another thing we're doing is we're using the ADI's ADU-CM355 SoC as our potentiostat. We've configured the ADU-CM355 to take high-resolution measurements on a single channel, as is necessary for our tests. as when we do our conoamperometry testing, we need to be able to sample data points every 15 to 20 microseconds. We've developed our system using the evaluation board of this SoC. I'm using a 3D printed box to demonstrate kind of the form factor for a reader that we ideally envision. So something with kind of this reset button and then a measure button, and then with wire connecting to the sensor probe. This is a further designed sensor clamp that we have that just uses a micro USB connection so that you can, quickly swap different holders that connect to the evaluation board through this box. And so this system here, this kind of handheld kind of reader with the sensing probe at the end, is currently being used by our collaborators in our COVID-19 animal study. As this is a CL3 lab space, so technicians are required to do the experiments for us, and this kind of gives them the form factor that we envision for a portable reader and probe. So here we have 4 chips with 2 different experimental conditions. 2 chips are BNP sensors, which will bind to the BNP protein, and 2 are BSA modified sensors, such that BNP won't be able to bind, and they act as a negative control. So here we take our chip for the experiment, put it into a clamp, as you can see here. This is then put onto our benchtop setup with the breakouts for all the working electrodes, and here you can see we're placing the chip such that the reference and counter electrodes are touching the fluid that is on the chip. And here we take another probe to connect it to one of our working electrodes. And here's the EDU-CM355 setup we're using, and it's connected to our computer with some in-house software for interacting with it. So here we're doing our measurements. So this system here, essentially that is our reset button, and all the prompts that are coming up on the Python script are things that can be put into an executable file. For now, we're running it on a Python console, just so you can see the visual display of the data on a graph. So there's the measure button that I just pressed, and we see the measurements taking place, and then there's the response. Once we've done all the measurements of the unbound probes, we then take 100 picograms per ml of their BMP protein and deposit it onto all of our sensors. And then we let it incubate for 10 minutes in this case. So here is our BNP sensor after 10 minutes. And as you can see, that's the unbound signal on the graph right there for comparison. And now we take a measurement after 10 minutes and we see protein detection. Next, we've put in our BSA modified chip, which we've added our positive protein into. Again, do a measurement with the unbound signal there for reference. And we take the measurement. Again, this is after 10 minutes, and we can see that the signal isn't significantly different, as we expect for a negative control.